Professor Robert Henry got his doctorate at La Trobe University and is currently a professor of innovation agriculture at the University of Queensland, Australia. For 10 years, Robert led the Queensland Alliance for Agriculture and Food Innovation, QAFI, whose mission is to significantly improve the competitiveness and sustainability of tropical and subtropical agriculture and food sectors through high-impact science. Throughout his career, Robert has always been a strong advocate of conservation of natural diversity and using genetics and genomics to understand and apply this natural diversity to improve agriculture for the world. This includes the genomics of crop wild relatives to learn from natural adaptation and molecular mechanisms of drought to enable us to continue to farm in a freshwater limited world. Robert has been a longtime JGI collaborator, including his work on Euclids, perennial sorghums, and of course, a co-PI on our sugarcane projects, as I mentioned, one of the most gruesome genomes ever to sequence. Today, he will be telling us about genomics of wild and domesticated plant diversity for crop improvement. If there are questions for Robert, please put them in the Q&A box and we'll have some time to answer them live. Thank you so much, Robert, for joining us so late to talk to us about crops and wild relatives.
Thank you, Jeremy, and I hope you can see my my screen. And
we can see your screen fine, Robert.
Great, thank you. Well, I've got September on my first slide because in the last hour we have started September here in Eastern Australia, and maybe that means that for people that are still back in August, I can tell you something about the future of plant genomics. What we see as the challenge really for the future of agriculture and food production is that it really needs to be, first of all, adapted to climate change. That is becoming the dominant requirement, and but also we need to, at the same time, continue to address the traditional consumer needs of taste and convenience, but we're adding to that a consumer's desire for healthy foods, sustainable foods, and ethically produced foods. And even more pressure for us in plant genomics is that we also need to have genomes that allow us to deliver non-food outcomes as we move to position plants to provide the carbon to replace sources of fossil carbon. And the responses that we have available to us in agriculture and adjusting to climate change, I classify into three categories. We can adapt, and that means that we need to modify the genomes to allow the plants to be productive in new environments. This is a challenging area, so one of the other options we've got is to move the crops, and we're seeing that happening on a large scale globally. But that doesn't completely avoid the need for genetics, because if we move to a place where the temperatures are the same, we may find the soils are different. And so we do still need to undertake major genetic adaptation. The third option, which is more radical, is to protect against the climate change. And that means we're going to move much more of our cropping into some sort of protected environment or vertical farming. And if we take this step, of course, we really do need plant genomics, because we need to design plants not to cope with a variable climate, but really to give that optimal performance in the environments that we're able to create. A key option that we have available to us, of course, is to capture much more of the biodiversity that exists in plants, and understanding the genomes of plants will really is a key way to enable this. I'd like to give examples today of how I see plant genome analysis contributing to that biological understanding that's a key to genetic improvement. And we do work at probably three levels. We work a lot with organella genomes, particularly chloroplast genomes, and that really tells us a lot about the scale of reticulate evolution that we see in plants. That's well beyond what we thought before we were able to analyze chloroplasts on such a large scale. The transcriptome is a particularly powerful tool for gene discovery for important traits, but it also allows us to look at things like allele-specific expression, which is something that's becoming more important to understand. But the nuclear genome really remains where the key focus needs to be. This is where we do most of the gene discovery, but also where we look at things like heterozygosity and increasingly the presence and absence of genes in different genotypes, and how that contributes to the performance of our crops. I'd like to take us back to the beginnings of agriculture to look at how genomics can both inform what happened in the past, but also, and in so doing, tell us something about where we need to go in the future. This is barley, wild barley, that I've photographed here in Aleppo, and this is it growing on the sides of the citadel before it was bombed some years ago. And this really is the plant that we believe was at the foundations of agriculture, the first plant that humans domesticated. And there's been many questions about that domestication. One that we've been able to solve by looking at the genome of large numbers of barley accessions is the origin of the Tibetan wild barleys. And here we were able to show that what had happened was that the domestication had happened in the Fertile Crescent in the Eastern Mediterranean, and barley had been brought across through India and up into Tibet as much as 5,000 years ago. And then that domesticated barley went through a bottleneck in going into a colder climate and then escaped into the wild. So we have now an explanation for the close relationship between the wild and domesticated barleys in Tibet, that it isn't one of domestication, it's rather of escape into the wild. These sorts of insights that come from looking at the genome of large numbers of individuals are really important in understanding how we go forward with plant improvement. We're now looking even further back. In Australia, we have continuous human habitation in areas for more than 65,000 years. And here we're beginning to look at the genomes of both the humans and the plants to try and understand how they've interacted over that time period, how plants have been modified by that human use, and what plants were used in the past that might be new opportunities for the future, especially for new climates in the future. A key focus for us is rice, because it's such an important crop, and what we're really looking to do is reinvent rice for the future, to have all those attributes that people want of it, but to have it in a way that we can produce it in the climates of the future. There is really huge diversity available to us in rice, and by looking at the genomes, we're able to understand how we can capture that. This map here depicts the chloroplast haplotypes we see in the wild rice progenitors across what I think of as the local area, from northern Australia up to northern China and across to India. Here we see quite a number of distinct groups, many of which have not been, or most of which have not been captured in domesticated rice at all. So there's a great diversity there that provides really new genetic opportunities that we haven't even begun to exploit in rice improvement. So by looking at the genomes, we can certainly start to understand the evolutionary relationships in rice and see the beginnings of domestication. We're focused on some wild populations in northern Australia that are unique in that they exist in an area where there's been no domestication. So we can consider these as representing real wild populations that haven't been impacted by gene flow from domesticated rice. And what we see here remarkably in different species is that there is hybridization going on in the wild population. So there's ongoing patterns of reticulate evolution in the wild that tells something about some of the events that have led to domestication and to the populations from which we've domesticated rice. Looking at the chloroplast of the chloroplasts that are available for the thousands of rice species that have been resequenced, we've made a remarkable discovery in that we've identified that there are only two functionally distinct chloroplast genotypes. There's quite a lot of variation in the chloroplast of domesticated rice, but only two functional haplotypes. So there's quite a number of amino acid differences in key proteins and even differences in the transfer RNA sequences, but they fall into just two functional haplotypes, suggesting very clearly two maternal domestications for rice. Now we know that there's evidence for multiple domestications of rice in the nuclear genome. And what the way I now present the argument is that rice has two mothers but many fathers. So from two domestications, maternal domestications, rice has been moved around the world and then interbred with local genotypes to produce quite distinct types. So the wild rices are effectively being domesticated locally, but these many different rices are all based upon two maternal domestications. And so there's a whole lot of other maternal genomes out there that have never been captured in the domesticated gene pool. And I think this examination really gives us a new perspective on crop domestication. And we're looking at many of these wild species that are interfertile with domesticated rice, have new disease resistances and novel health and nutritional traits that we can now begin to capture. A good example of the sort of opportunity that the wild species provide is shown here. The grain B is a wild accession where E is a domesticate, and you can see we have the ability to access much larger grains in the wild, and the genetics of this is something that we could readily incorporate into domesticated rice with potential with larger seeds for yield enhancement. The other area that's quite exciting looking at the wild rice is that the starch properties and general properties of the grain suggest that many of these wild rices will be more slowly digested and will have health advantages where in communities where there's large consumption of rice. And this has been an objective for a long time, but the mutations that have been possible in domesticated rice have led to rice with undesirable textures and tastes. What we're now finding is rice that might be healthy but also taste good in the wild, and bringing that into domesticated rice is likely to have a very important impact on the health of communities that rely on rice as a major food. Genomics of rice has really given us a lot of insights into important traits. None may be more important than fragrance. This single gene doubles the value of rice, and when the rice genome was first available to us, we very quickly discovered this gene because we had flanking markers. And this gene we then immediately identified resulted in a loss of performance because it was a gene that impaired a stress response pathway, the metabolism of proline. So we solved the mystery of why fragrant rices were low yielding. Subsequent studies in other plants are now showing us pathways that will allow us to produce this fragrance without paying the penalty of the loss of performance or less of stress tolerance that results from disabling this pathway. So comparative genomics, I think, will lead the way in allowing us to produce rices with the qualities that humans want. Fragrance is attractive to humans as 2-acetol-1-pyrroline at 10 parts per billion, but it results in a great loss of performance of the plant. We now have prospect of bringing the performance back into these desirable types. I've talked about rice, but wheat, of course, is one of the other really big cereals in the world, much more widely grown, much lower yielding in much drier environments. By wheat, looking at genomic approaches, particularly transcriptomics, has allowed us to discover genes that might improve grain yield, but more importantly, improve the whole productivity of the system. We've identified the genes that determine how much flour we get from a ton of wheat and how much bread we can make from a ton of flour. So by bringing all of this genetics together, we can get much more food from the same amount of grain. And to illustrate, I think, how genomics has provided these insights, I'll cite this protein. We call this the bread-making gene. This is the protein we predicted encodes We discovered this by looking at the transcriptome of wheats with different bread characteristics. This is a very small protein, had never been detected as a protein before, was not annotated in the genome because this is a very small gene, but the transcriptome showed us that this was one of the most abundant transcripts and present only in wheats that gave high loaf volumes. So this, we believe, is a protein that cross-links the large proteins that the researchers have been focused on for the last 100 years and provides a mechanism for supporting a risen bread product. And this is really something that's could only easily be discovered by looking in detail at the transcriptome. Similarly, the transcriptome has revealed the genes that determine how much flour we get from wheat. And what we've found is that human selection has involved reducing the expression of cell adhesion proteins that hold the seed together. So simply by weakening the grain, we get more flour. And this was a great challenge to wheat breeders because we had genes in different places in the genome. But what this shows is that they all had the same function. They're all associated with cell adhesion. This understanding of the mechanism of control of the yield of flour allows us to produce more food from the same amount of grain. Similarly, the wheat gene pool shows great diversity in response to temperature, and adapting wheat to more extreme temperatures is a really key global objective. By looking at wheats that have been selected in tropical environments, remarkably, we've found some wheats with extreme tolerance to heat. So even though these genes, again, are not widespread in the wheat gene pool, they are in domesticated wheat. So by understanding the genes that are expressed in response to heat, we really have an opportunity to breed much more heat stress tolerant wheats for the future. And in this case, we don't need necessarily to go outside the domesticated gene pool. The variation seems to be there. I'd like to talk about sorghum. This is another species that is of particular interest, and I know that JGI has been very involved with sorghum genome work. Our interest is because much of the diversity at the genus level is in Northern Australia. In fact, the bulk of the species are found here. And so we're now beginning to understand how we can utilize that greater diversity to support sorghum improvement. We have species with many different characteristics that have real potential for enhancing sorghum as a crop for food, energy, and all the other things that we're thinking of using it for. We've been working with JBay and JGI in exploring the genomes of some of these species. We've also started another program that I think is an important complement. The Carlsberg Foundation is funding us to develop a very large population of mutant sorghums. This is some of the first material in the field. We're aiming this year to have 500,000 mutants of sorghum. So what we'll effectively have is mutants in every gene in sorghum that will be available as DNA and as seed. And this will allow gene discovery and rapid deployment of new genetics in sorghum. This is particularly designed to satisfy the needs of the European market, where gene editing and gene modification are still difficult to commercialize. Mutation is much more acceptable. So here is a way of reverse engineering some of those genotypes. And by looking at the genus, we're now understanding that there is substantial reticulate evolution going on here, and that there are many close relatives of domesticated sorghum that haven't been explored, that have characteristics that we can bring into production. If we, I'd like to turn to sugarcane, because in many ways sugarcane is polyploid sorghum. They're each other's closest relative amongst domesticated plants, And sugarcane is the biggest crop in the world, one of the last crops for which we've got a good quality, are likely to have a good quality reference genome because of its high polyploidy. What I'd like to share with you today is some recent work we've done looking at allele-specific expression. Here, where we've got a species with a very high level of ploidy, 12 ploid on average, we have the opportunity to understand at a different level allele-specific expression. So here I'd like to, these figures show 5 different tissues where we've plotted gene expression but allele, the expression of the allele versus the abundance of the allele or the copy number of the allele in the genome. And maybe to understand this well, you need to look up in the top right-hand corner. You'll see a very high level of expression of bases that are not polymorphic in the genome. That's what you'd expect. But if you come down that line, I hope you can see that there are a series of discrete patches. So what we're seeing is expression of 1, 2, 3, 4, up to 12 copies of the gene. And what we're seeing is that expression is unbiased. So it's remarkable that here we're able to see discrete levels of expression, that we can separate 3 copies from 4 copies, from 5 copies, all the way up to 12 copies, and that probably 80% of the genes are being expressed, or 80% of the polymorphisms in the genome are expressed in an unbiased way. So it's the first time we've really been able to do that quantitatively. Here we've sequenced these 10 gig genomes to 100x, so we've got a lot of data to support this analysis. But we do see a smaller number of genes that show completely biased expression, regardless of copy number, and these are the ones that are along the top of these figures. These are predominantly genes associated with pest and disease resistance that have been under strong selection, both pre and post domestication. So what we can now do by deep sequencing of both the transcriptome and the genome is come to a greater understanding of which genes are preferentially expressed and how a polyploid system operates in terms of differential expression. So we're really interested in how, of course, we can redesign crops like sugarcane particularly, but also things like sorghum to take this role as alternative sources of carbon. Our focus has turned from, as I think in many places, has turned from using these things to produce fuels to thinking much more about the biomaterials, the other requirements for fossil carbon, as we move more to seeing the future of transportation involving electrification. I'd also just like to mention the importance of pulses and protein crops. The world is wanting to develop more sustainable sources of protein, and what we have is a great diversity of legumes in the wild that haven't been domesticated. So there are many new crops that could be that climate resilient protein source. And what we really need to do is a lot of genomics to understand the utility of these species and how they can be either used in producing new crops or used as sources of genetic diversity for existing protein crops. One that we're focused on at the moment is the pigeon pea, where we can see great diversity in the wild that we're just beginning to capture, including resistant to the major insect pests of this species. We're also working with many of the high-value horticultural products because they're now at the stage where a genomics platform can enable acceleration of the breeding of many of these tree crops that have long life cycles. So making the right selection is really important because it the trees are with us for a long time. Citrus is one example, and here we have on the left Citrus australasica. We got the first assemblies of that this morning, and I can say that it was quite surprising to learn that it's a tetraploid, and I think it's telling that we're learning about the ploidy of species from their genome sequence rather than from other techniques, because that really is the easiest way to understand a completely unknown species. We also learn the number of chromosomes and so forth very readily now from an assembly of the genome. And so we're now beginning to sequence very many wild citrus, for example, that have resistance to the major diseases that are becoming problems globally for citrus production. The Macadamia is a local species here that was actually taken to Hawaii and domesticated there. We're using this as a model, and we're putting this whole industry on a genomic basis by really sequencing everything. We're sequencing all known varieties, all of the species. We're providing high quality reference genomes, and we're sequencing all of the diversity in the wild, in some cases for the rare individuals, now every individual of the species. And this is the rare species where there's only a couple of hundred. We hope to have good deep sequencing for every individual. And we're using this as a model to test genome sequencing and assembly techniques. And it's fair to say that this is something that we see as continuing to advance at a substantial rate. We're seeing real improvements both in the sequencing and the assembly, and the lengths of the contigs and the quality of the assemblies dramatically improving for this and all the other systems that we're applying it to. And I suppose the point that we've got to at the moment is we're starting to see assemblies that provide us a single contig per chromosome, and that is more common than not, so that we find that for many assemblies, we now expect that most chromosomes will correspond to a single contig. So we no longer may need things like high C if we can really assemble long reads to the full length chromosome. And this is something that's really only enabled by very accurate and deep sequencing and good assembly tools. But this promises, I think, a sort of new era in plant genomics. So we're looking to apply this to do comparative genomics and support key gene discovery in many of these crops. One of the applications that's immediately bearing outcomes is looking at cyanogenic glucoside formation in plants. Remarkably, the work that we're doing is continuing to confirm that this is something that's been selected for in domesticated plants. We always thought that the wild plants would have had the toxins and that we selected against them, but remarkably, we seem to have selected for the formation of cyanide in the domestication of a number of key plant species. And comparative genomics is going to allow us to understand how and why, but it may be that in the agricultural context, the plants need greater protection in a monoculture against pests and diseases than they actually do in the wild. And so there's stronger selection for the production of these toxic compounds. Coffee is a really interesting example, and it tells us a number of things. Having a robuster coffee genome is being followed by better and better Arabica coffee genomes, and we're looking at much of the wild diversity, particularly in thinking about adapting coffee to climate change. Really,
I'd like to really just finish by emphasising that I think this is actually an opportunity to promote awareness of climate issues.
So that the environment and coffee quality is really important interaction. We've looked at the transcriptome of coffee beans from different levels in the canopy and shown that really the high quality coffee from high altitudes is associated with slow development of the berry. I've had a lot of media interaction because I think Kew suggested that coffee in the wild would become extinct because of climate change, and we're going to have problems producing high quality coffee. And it's interesting that this has created a lot of public concern about climate change. And I really think that what this provides is an opportunity for genomics to inform the public. What we see is that many people that may be not concerned about climate change are quite concerned about coffee. And if climate change threatens their coffee, maybe they'll get more interested in doing something about climate change. It's a really interesting system, and there is a real interaction here. We're also working on the genomics of a lot of species that are important, producing compounds that are used widely in medical applications. And we now are in a position where we can really edit the genomes of these plants to really enhance the levels of the bioactive compounds that are used in pharmaceuticals. One of the recent discoveries I think is really interesting that I wanted to share is that we've looked at sequencing dioecious plants, that's plants with separate male and female flowers, and what we've found is a huge divergence in Juboisia, in jojoba. This is a plant, of course, from North America, and what we see is a great dimorphism between the male and the female. And when we've sequenced the genomes, and remarkably, it's the first time anyone seems to have sequenced the whole genome of a male and female plant, what we see is a massive insertion in the Y chromosome. The difference in these genomes is enormous compared to what we see between males and females in animals, for example. We've got 2 5 megabase insertions in the Y chromosome, and we've got hundreds of genes that are male specific, and many genes that are in the X chromosome in the female specific regions. What this shows is that there's been long divergence and separate adaptation of these two sexes to different environmental niches, and that the stress responses of these plants are very different. The males and females will have different survival in climate change, and this suggests the precarious nature of these plants in that one of the genders may, one of the sexes may actually cope with climate change and the other may not. So it really is a remarkable divergence that we're seeing in the sex chromosomes. I wanted to also mention that woody biomass is something that's really important that we study more. We have good genomes of the eucalypts, and through work at JGI, we now have the Corymbia genome. The Corymbias diverged about 60 million years ago and have gone through long periods of parallel evolution. But these woody species are really important for biomass production. And one of the places that we're now going is doing comparative genomics in these species to understand really important traits. For example, here we're looking at trees that are completely different in size, but are very similar genetically. We should be able to understand the genetic control of plant size in an absolute sense by the comparative genomics that we can do in these tree species. So to summarize, really, I see the great opportunity to capture more diversity for a very wide range of major crops, and that genomics is the key tool that allows us to do that. Here in Australia, we've got a top-down approach. We're looking to sequence each of the families of plants, then each of the genera, and in the long term, each of the species. We have about 10% of global flora, so this is going to take a little while, but the program is in place. We're taking a systematic approach. And the other thing that we're doing is looking at all the threatened species. This is one that we're sequencing, where we can look at a tree here where there's only 99 individuals of the species, and we can sequence everything, all of the individuals, and really manage well these particularly endangered species by having a full knowledge of the diversity that exists. So crop wild relatives and new crops are two different approaches. I think we can capture more diversity for our existing crops from wild relatives, but we need to think about completely new crops for the future. And here's an example of a grass that we're looking to domesticate as an alternative to rice. It's a grass that grows in cool climates, so this might allow us to produce a rice-like plant that doesn't have to be grown in the tropics and doesn't need a lot of water. And with what we know about the genes of domestication, we can really now think about doing that. We can domesticate completely new crops. So I just end by acknowledging all the organisations we're collaborating with in our genomics work, and thank all the many collaborators, students and postdocs that are working on these projects, and leave it there and welcome any questions. Thank you.
Thank you, Robert. Thank you. That was a most fantastic, very broad presentation of what you guys are doing to try to select and adapt moving forward with crops in the future. There are a few questions piling up. Please feel free to put additional things in Q&A. And so I thought maybe we'd start with a relatively easy one here. Do the two types of rice based on the chloroplast genomes have unique phenotypes? And do those chloroplast genomes tell you anything about the origin and domestication of rice?
No, it seems that there may well be a genetic cost or value associated with these two distinct chloroplasts, but due to the way that rices have been developed, it seems that there's no correlation. For example, Indica and Japonica rices are found with both chloroplast types. So most of the subgroups within rice are found with both chloroplast types. So we haven't yet got the data on what the impact of those differences is, but it may do something like influence response to light, because some of the genes there are probably able to make the plant more shade tolerant, for example. So there may be understanding that may actually provide an advantage in rice breeding.
So there may be an advantage to having different chloroplasts in different kinds of nuclear backgrounds.
Yes, I think so, and we really haven't begun to explore that. We've just really opened the question by making this observation that there's that big difference.
Great, thank you. I'm moving to something a little bit more complicated. So you talked about climate change and potential solutions for plants, and you did mention genetically engineering crops or organisms as a potential solution. But there's also biocontainment issues around that, and thinking about how do you mentioned, I think, acceptance from the perspective of end users in European Union. Could you share some more perspectives on what you think about how you would actually use engineering practically to adapt the crops to climate change?
Yeah, so I could cite the example of the path we're taking at the moment with rice. So with rice, we're developing the ability to use CRISPR-Cas in both domesticated rice and in the wild species in parallel. So what we're able to think about doing is editing novel alleles we find in the wild into a domesticated background to understand the value of those genes, but equally to take domestication genes from domesticated rice and edit them into a wild rice to domesticate it, in effect. So I think we can follow both those paths using those sorts of tools. So I think there's real opportunity to do that in much higher throughput. We can really start to look at lots of genes now using these techniques in something like rice, which can be edited in high throughput. And so we're hoping that being able to move in both directions between the wild and domesticated will give us the power to really discover the way forward.
And what do you see as the sort of major targets for crop adaptation in the climate change world?
Well, it's adaptation to the environments we actually have available for crop production, and we have to start by trying to understand what they are. I talked about the fact that we are going to be moving agriculture. It's moving north and south away from the equator. It's moving to higher altitudes. All those things are happening. But the biggest adaptation I believe, is going to be towards greater pest and disease resistance. We've looked in wild populations at the variation in the genome across temperature gradients, for example, in a wild plant species, and where we see the biggest variation is in the pest and disease resistance genes. So that leads us to suspect that the need is not so much adaptation to higher temperature per se, but to the greater range of pests and diseases that we'll find in those warmer temperatures. So I think it is that insect resistance, fungal disease resistance, they are going to be the key things that have to move to cope with a much more hostile environment for the plant.
Yeah, yes. I would also mention weed pressures and the interaction with local plants
Yes.
in that regard, right? As you kind of think about this, though, are there different strategies for different parts of the world? I mean, a difference for like major commodity crops versus sort of minor crops?
Yes, I think that very high value crops can consider the option, of course, of protected cropping or vertical farming. But and what really will determine how far down the list of value we can come with those sorts of technologies will depend upon how high we can get the performance under those conditions, because we do have the opportunity to have much higher performance if we control the environment. So that may allow us to produce crops that are probably lower in value than we're currently considering in a protected environment, if we can really get that exceptional yield to justify the economic investment in the environmental control. But yeah, all of those factors, there's a lot of people trying to understand which crops and how to take them down the various paths for adaptation.
It's really a balance. Between sort of inputs and outputs, right? Because in the current environment, it's a significant investment in inputs to
Yes.
get those yields out the other side, as
Yes.
opposed to kind of planting them out in the ground where a lot of stuff gets taken care of for you.
Yeah. So you've got to have the value to justify the investment. Yeah.
Great. So moving on a little bit to talking about domesticating, new plants, which is something I'm really excited about also, and using accelerated genomic techniques to be able to really, bring the domestication lessons we are learning from these historical domestications into new plants. Do you anticipate that there will be new allergy or toxicity issues as we domesticate new plants over time?
Yes. I think. Well, I'm sure there will be, but they need, and there'll be things that I think we can, in many cases, anticipate. Not always, but I think in many cases we will be able to anticipate some of those problems, because we already know about the allergenicity of certain groups of proteins that we're likely to find in particular plant families, for example. But that will be something that I think can be tested and needs to be tested as part of going into anything that's completely new. What we're doing at the moment is looking back at some of the plants that might have been used by humans tens of thousands of years ago to see if there's any species that are overlooked that we could bring back, and maybe some of those have been tested by humans long ago.
Right. So the sort of early domesticated species that have fallen out of modern farming.
Yes. And I don't think we know about all of them yet.
Right. I think there's sort of rich history here, also in Central, North, and South America too.
Yes.
So kind of pre-domestication species that were domesticated and used in farming applications by the original inhabitants. And so those are really interesting kind of targets for that.
Yes, it's almost best known in North America, I think, but it's probably something that's a worldwide opportunity.
Great, thank you. And then I think we've got time to get one more science question in here. When you're looking at sex chromosomes in these dioecious species, and asking, in your case, this is what did you learn about the gene variation that's linked to these different sex chromosomes and different sexes?
Well, we found in jojoba that very large numbers of genes that you'd expect, genes associated with flower development, the pollen, all the organs that are associated with sexual differentiation. But what we also found was very large numbers of genes associated with response to stress. So these males and females have developed different approaches, and really they're under different selection for reproductive success. The males need to invest far less in pollen than the females do in seed. So there are different criteria for survival and reproductive success that have driven them down quite divergent evolutionary paths. something to an extent beyond what we thought would be the case. 400 male-specific genes that are just not in the female. And most of those are sort of transcription factors and things that are regulating response to stress in a different way in the male and in the female. And that might provide more lessons about how plants do adapt, of course, and that we can use. But remarkable divergence there.
Yeah, I think plants are interesting in that sex determination has developed multiple times,
yes,
depending on where you are across the phylogeny or even within the genus level. And so there's lots of kinds of ways to mix and match that splitting into two separate sexes that come back together again. And so we're starting to see really interesting results also looking at sequencing the male and female and asking what's really the variation, which genes are linked to those, because not only, as you mentioned, there's those genes that are just different between the male and female chromosomes or regions, but there's also lots of genes that are linked to those that get driven by those. And so there can be thousands of genes in the plants which are linked directly to.
There are large number of genes that it shows sex-specific expression, even though they are present in both the male and female. That's because there's regulatory genes that are sex-specific that are driving very different patterns of expression. Exactly.
Yeah, that's a really great point. And I think we'll start to learn a little bit more about how that works in these species as we accumulate some more genomes and be able to do the kinds of direct comparisons that you're talking about.
Yes,
that's great. Okay, well, I want to thank you, Robert, again for joining us and giving us such a interesting, provocative talk about the future. And we all need to be thinking, from the plant and environment side about how do we adapt agriculture to this changing world, and how do we reduce the impact of agriculture, on its contributions to the changing climate and greenhouse gases. So I really appreciate it, and I want to thank you again for coming today.
Thank you.
Okay.